Hello, this is a self-structure tutorial presented by SchrDbb with reference Atlas of Histology and Functional Clinical Calculation by Dongmei Cui. So we're going to visualize the cell structures in this tutorial. So the tutorial is going to be divided. The cytology tutorial is going to be divided into different parts. At the primary part, you're going to have an introduction to visualize the different elements that will be seen inside our tutorial. Then the second part, we're going to show all the different elements inside the tutorial. Is it clear? So you will start now with the basic component of the cells. So the basic component of the cells are going to be divided into three. We have the nucleus, we have the cytoplasm, and we have the cell surface or the cell membrane. So now the first thing is the nucleus. So the nucleus is made up of different parts. It is made up of the nuclear envelope, which is a double membrane structure. It is made up of nuclear pore, which are gaps in the nuclear envelope. It is made up of heterochromatin, which is a condensation of chromosomes. It is made up of euchromatin, which is not condensed. So this one is like chromosomes. So, and it is made up of a nucleolus, and nucleolus is a spherical structure located inside the nucleus and generating ribosomes. Now the next thing to visualize will be the cytoplasm. So the cytoplasm is made up of different elements. The first thing is the ribosome. So what is the ribosome? The ribosome is the cell structure helping producing proteins. Polyribosome is just a chain of ribosomes. The next one is the rough endoplasmic reticulum. The rough endoplasmic reticulum are flattened membranes delineated cisternae. So cisternae are just tubules which have a certain pattern. So those are cisternae. So, and the rough endoplasmic reticulum has ribosomes or polyribosomes attached on the surface. The next thing is the Golgi complex. So this Golgi complex is also made up of flattened membrane cisternae, and they are located and they are at the periphery of those flattened membranous cisternae. We have usually vesicles. The next thing is the secretory vesicles. So as I said, the secretory vesicles are directly connected to the Golgi complex, where the channeling of materials inside the cells is going to occur. The tracking of material inside the cell are going to occur. Then the next one is the lysosome. The lysosome is contains the enzyme lysozyme, which is used in the lysis of the endocytic components. Then we have other element of the vesicles. After that, we have the smooth endoplasmic reticulum, which is just made up of a labyrinth of tubules. So the supraendoplasmic reticulum is not cisternae. It means that it is not having a particular pattern. It's a tubule without a particular pattern. It can be branch, it can be Y-shaped, or it can be the rest. Then we have the mitochondrion. The mitochondrion is one of the simplest organs in the nucleus to identify. Why? Because it is made up of a double membrane, and the inner membrane is made up of cristae, and cristae is just plicated inner cell membrane. Then the third element is going to be the cell surface. So at the cell surface, you have the cilia. The cilia are just motor elements or motor structures of the cell membrane. We have microvilli. Microvilli are elements of the structures of cell membrane which are used to increase the surface area for reabsorption. We have stereocilia. The stereocilia is just a long microvilli, as easy as that. Then we have how the question I want to ask ourselves is certain structures, certain tissues in the body are connected to cells of the soul. Certain tissues in the body are connected to each other. So we have certain structures which are used in their connection. So example of structures using connection is zonular occludens, or it's also called the tight junction, which is located at the apical most part of a cell. Then you have the zonular adherens, which is located just inferior to the zonular occludens and attaches to the actin filaments. We have the macular adherens. The macular adherens is also called the desmosome, and it is similar to zonular adherens, located just inferior to zonular adherens, and it's also connected by intermediate filaments. Then the next one is the gap junction. The gap junction is also called the nexus junction, and it is connected by proteins called connexons. Then at the at the basal most part of the cell, usually located in the epithelial cells, we have the hemidesmosomes, which are just similar to the desmosome, just that they don't have a mirror image of the plaques and the mirror image of the intermediate filaments connected to each other. So we have the basal lamina. The basal lamina is just in epithelial cells is going to be inferior, is one part of the basement membrane and can be seen only the ultrastructure of the cell. Then the last one that you're going to see, cell structure components, is cytoskeleton. And the cytoskeleton is made up of different filaments. You have the actin filament, which is much more located at the periphery and it is used to maintain the structure of the cell. Then we have the intermediate filament. The intermediate filament is located just inner to the actin filament and also used in the maintenance of the structure of the cells. The microtubule is a tubular shaped structure, and it is used in tracking the movement of particles or different cell components inside the cell. And the last one is centrosome. The centrosome is cell structure which is made up of a pair of centriole, and that pair of centriole is used in the movement is used in the to make the microtubule to have a synchronous movement. Now let's go again. Actin filament is the smallest, then you have the intermediate filament which is slightly larger than actin filament, then microtubule is the largest. So in order for you to know the different diameters, is actin filament is 6 nanometers, intermediate filament is 6 times 2, that's 12 nanometers, and microtubules is 12 times 2, that is 24 or 25 nanometers in diameter. Now the next thing to visualize are the different functions. So the 50 different functions, I'm not going to speak about that. Now this is the ultrastructure, a diagrammatic representation of the ultrastructure of a cell. As you can see here, you have the cilium. Inside the cilium we have microtubules which make up the cilium. At the end of the cilium we have the basal bodies. Then still on the cell surface you have the microvillus. The microvillus is made up of the actin filament, and at the end of the actin filament down here part it is called the terminal web. Then here we have the secretory vesicle, which is just going to be produced by the Golgi apparatus here. And the Golgi apparatus, as you can see, it is having most of the time a U shape. It is made up of cisternae, most of the time a U shape. Now at the U shape we have two different parts. We have a concave part and we have a convex part, and you have a concave part. The concave part is the mature-most part, is the part which is most matured, and the convex part is the part which is most immature. That is the vesicles which are coming here are not yet modified. Is it clear? So the next structure that you can see microtubule that is used in maintaining the cytoskeleton. We have the rough endoplasmic reticulum here. These are free rough endoplasmic reticulum. These are rough endoplasmic reticulum which is directly connected to the nuclear envelope. Now as you can see, the rough endoplasmic reticulum surface is containing the ribosomes. Now we have ribosome here. Is a single cell structure that is used in maintaining producing proteins. Then you have polyribosome, which is a chain. We have the lysosome, which contains lysozyme. We have here the hemidesmosome, and it is connected here by the intermediate filaments. As you can see on this other part here, we also have here your desmosomes, and the desmosome is also connected to the intermediate filament. Just that the desmosome is connected to the intermediate filament, and then the other cell, which is the mirror image of this other cell, is also having a plaque here and intermediate filaments. So this is the hemidesmosome, but in the case of hemidesmosome, we don't have the mirror image substance. At the inferior part of the cell, this cell is mostly an epithelial cell, so you are going to have a basal lamina. Now, the next structure of the cell is going to be smooth endoplasmic reticulum. So the endoplasmic reticulum is not actually a cystine pattern, it's much more a branch tubular pattern, as you can see here. It is a tubule, but it is not having a linear pattern like in a cystine, it's having a branch shape. The mitochondria is made up of two membranes, the outer membrane and the inner membrane, which is divided, which can be intertwined by like a plique, as you can see here. Now, the next one is the cells, the surface of the cell. The cell surface is made up of the zonular occludens, the zonular adherents, the zonular adherents attached to the actin filament. Zonular occludens, no particular protein. Zonular adherents, intermediate filament in the actin filament. The desmosomes is attached, or the macular adherents attached to the intermediate filament, and the gap junction attached by the connexon protein. So we are finished from this view. Now, this is the electromagnet view of the ultrastructure of a cell. As you can see here, this is our nucleus. So inside the nucleus, you have the nuclear envelope, as you can see here. The nuclear envelope. After the nuclear envelope, you have also here the heterochromatin. This is the nucleolus. This is the euchromatin. Now, the next thing you can visualize here, these structures here, which are round like that, are the mitochondria. As you can see here, mitochondria. So the mitochondria, as you can see, if you visualize very well, you see some certain lines inside the mitochondria. These are the plique, the cristae of the mitochondria that make it the structure to be identifiable. Now, this is a lysosome. When you see a large vesicle, which is usually dark in color, it is just a lysosome, and this one is called secondary lysosome because the material is already being digested. Then usually the parts which is all just is to fill the fluid of the cytoplasm is much more made up of the smooth endoplasmic reticulum. Smooth endoplasmic reticulum, you see tubules which are branch in shape. When you see tubules which are branch, just know that it's smooth endoplasmic reticulum. But in this case, when you see tubules which are elongated to form a cystine, and at the apical part, you see some dots, dark dots, that's a rough endoplasmic reticulum. And at the terminal portion here, you see the cell membrane. The cell membrane is not actually smooth as it is seen in the upper diagram there. It is having some interruptions, as you can see here. So this is cell membrane, also called the plasma lemma. Now, this is the second ultrastructure. So in the second ultrastructure, what are you going to visualize? So this is still the nucleus with the nuclear envelope, but then you also have the nuclear envelope also has the cystine of the nuclear envelope. Now, the cystine of the nuclear envelope also have the nuclear pores. The nuclear pores is the communication area between the nucleus and the cytoplasm. Then this is our heterochromatin, this is our euchromatin, this is our nucleolus. Now, the next thing is the nuclear envelope. The nuclear envelope is going to be divided into two. It is divided into the outer nuclear membrane and the inner nuclear membrane. Both of them have pores, and those pores have structures which are used in regulating their entry. This is a pores here. They are used in regulating the entry and the exit of the different elements, which different particles which communicate with the nucleus and the cytoplasm, and that's structure is going to be called the diaphragm. So the diaphragm is just a structure that is going to be used in regulating the movement of particles into and out of the nucleus. So this is again the rough endoplasmic reticulum. The rough endoplasmic reticulum, we see that it is a tubular system, and then at the apical most part of it you have ribosomes. So you see here tubular system, apical most part of it you have ribosomes. Here also tubular system, tubular system, tubular system, and then you see ribosomes. So this is a cell. As you can see from this structure of this cell, you can already guess that this cell is producing much proteins, and it's a cell which is producing high. It's a secretory cell that produces much proteins, more than steroids. But if you see more of smooth endoplasmic reticulum than rough endoplasmic reticulum, you see that it's a cell that produces more steroids than protein structures. So this can be an endocrine cell. So the next thing to visualize is going to be this is another portion of the Golgi. So but here you much more see Golgi apparatus, and the magnification is much more increased. So you see the mitochondria here. This is the mitochondrion. Here you can see the inner cristae, which are plicated, as you can see here. Now this is our Golgi complex. So the Golgi complex, you can actually see the U shape of the Golgi apparatus here, and this U shape has both the concave is the part that enters, while the convex is the part that goes out. So the concave end is the mature most end. So it means that what is inside here, this portion here, is going to be the secretory vesicles, which are already formed for being mature, can be tracked in different part of the body cell, or moved to different part of the cell. While this other end here is the convex end, and this one which are entering here are the immature vesicles, which are coming into to be formed and to be processed by the Golgi complex. So now you can see also a cistern here, and then at the surface of the cistern you have ribosomes. So this is a rough endoplasmic reticulum. And then this you see a round structure, a vesicle, which is dark in color. When it is dark in color, just know that it's a lysosome. But if it is much more paler than this, it's going to be secondary lysosome, because the material is being digested. Then you see the double membrane of the mitochondria are the same. This is the central circular DNA of the mitochondria, as you can see here. So these are the plicated part of the mitochondria and all that. So the next structure, so in this structure view, we're much more going to be concentrated on the rough endoplasmic reticulum and the Golgi bodies. So the rough endoplasmic reticulum, as you can visualize here, it is made up of cisternae, as you can visualize it, and then ribosomes at the topmost part of the rough endoplasmic reticulum. You can actually visualize it here. So it is made up of branching cisternae, tubular structures, or we can just call it labyrinths if you want. Now this is the Golgi apparatus here. The Golgi apparatus, you see it is here, this structure is not actually having the typical U shape that we know, but as you can see, it has a small turn in this direction here. So this direction just tells us there that this part here is the immature part, and this part here is the more mature part. So you have a small U-turn, though it is not actually very, very conspicuous. And then these are the mature vesicles here. Now the next structures here, when you see tubules which are not actually cisternae, tubules which are just irregular in diameter, just like this, it is much more of a smooth endoplasmic reticulum. And smooth endoplasmic reticulum are used in the production of hormones which are not protein structures. They are hormones which are much more going to be fatty or philic to fat. So example, we have steroids. It can be used in the production of carbohydrates. It can be used so it has different functions. So you mostly use in the production of steroids, and also used in the changing of to processing of also vesicles inside the cell, while the rough endoplasmic reticulum is much more used in the production of proteins. So this is the mitochondria. We've already seen this is a lysosome. We've seen that, and this is a plasma level. When you see small lines, when you see lines like this, lines, you see lines like this inside the cell. These lines are much more going to be are going to be filaments. When you see a single line inside the cell, it's going to be the intermediate filament. But when you see a tube, like a tube line, like these lines that you see here, these are much more microtubules. But when you see single lines like here, it is much more intermediate filaments. Now the question you can ask me is, where are the actin filaments? As you have already seen before, actin filaments are much more located at the cell surface. So this is one cell surface. The lines which are here, located here, are the which are closer to the cell surface, are the actin filaments. So you don't see actin filaments far inside, you instead see it outside. So it is much more the periphery of the cell. So this is the plasma lemma, and these lines are the actin filaments. Now this is the structural view at the cell surface. So this is the junction between two cells. So at the junction of between two cells, we said that we had the adhesions between the two cells. So at the topmost part, we have the zonular occludens, which is the tight junction. You have the zonular adherents. Then at lower part, we have the macular adherents of the desmosome. So you have the plaque here. Now you see here, the zonular adherence is going to be attached to the different actin filaments. So this part here is the actin filaments with the terminal web. This part here now, which is the inferiormost part, we are going to have intermittent filaments here. So you have intermittent filaments, you have to that the macular adherence is going to attach. Now the question you can ask me is, what is this? All these are actually the cross section of a microtubule. I've initially told you that a cross section of a microvilli. Sorry, I've initially told you that the microvilli are made up of multiple actin filaments. So you have actin filaments, multiple actin filaments, and you can see this microvilli is circular, and then you have multiple actin filaments inside, and then at the lowermost part of that microvilli, you have a terminal web. As you have seen, if you want to go, you could just scroll up back to, you can just play back the video to see the terminal web in the longitudinal section of the cell. Now this is still to visualize the cytoskeleton. As I've initially told you, when you see a tube, as you can see here, this tube is a small tube. It is a more of microtubule. But when you see a line, just a line, a single line, it is more of an intermediate filament. This is still mitochondria, and these are the synaptic vesicles, because this one is a cell structure of a neuron. And when you have microtubules inside, and the intermediate filaments inside the neuron, is going to be specially called neurofilaments. So now this is the cell structure now on a light microscope. So this is a very huge cell, so on a light microscope, but the problem is that the light as you can see, you cannot actually you don't have a very good resolution. You cannot very define the different cells. Are you seeing? This is because the light microscope is not having a good resolution. The resolution of light microscope is only 0.2 micrometers. It means that let's go back a bit to physics and define resolution. The resolution is the distance, they mark the smallest distance to which you can the smallest the ability to, let me just define it in a clearer view. A resolution is defined as the ability of a material to distinguish two distinct points of the different part of a body. So that's the definition of resolution. Are you getting? The ability of a material to distinguish between two structures of a cell. That's the resolution. Now, if I tell you that the resolution of a light microscope is 0.2 micrometers, it means that what? It means that the light microscope can only distinguish cells with a maximal distance. It can distinguish two parts of the cells clearly which with a maximal distance of 0.2 micrometers. You see, that's actually it's a small diameter, it's a small distance, but it is not useful in microscopy, because microscopy, if you want to visualize like the mitochondrion is very small compared to the 0.2 micrometers. If you want to visualize the smooth endoplasmic reticulum is smaller than the 0.2 micrometers. So it means that you cannot see smooth endoplasmic reticulum in the light microscope. You cannot see rough endoplasmic reticulum in the light microscope. All the cellular structure which are smaller than the 0.2 micrometers, you cannot see them on a light microscope. That's why light microscope is not actually very used in cytology, but it's going to be much more used in histology, as we are going to see later. Now this is the secretory granules. These are the secretory granules here. As you can see, you have the nucleus with the nucleolus. So these are cells which are used in secretions. So we have the secretory cells. So these are columnar cells. At the uppermost part here, the secretory granule. At the lowermost part, you have the nucleus and the nucleolus. The nucleus and the nucleolus, lowermost part. So these are different cells, and these cells are attached in such a way that they are glandular structures. So they are secreting directly into the tubulus. So these are tubular glands, as we are going to see in the gland structure. Now this is, as you can see here, you see the light microscope. When you are going far down, you are not capable of distinguishing the structures very well. So this is a cell that you have magnified a lot. That's 780 magnification in order to visualize the nucleus. You see, you can actually see the nucleolus. You can actually see the euchromatin. You see that this is the euchromatin. You can say that this is the nuclear envelope. Actually, you can see it, but the clarity is not good. You are capable of the magnification can never end with a light or any microwave. Magnification can never end. If you want to take your phone, you can just try with your phone. You can continuously magnify with your phone, but the more you magnify, the lesser you can distinguish between the 2 points of that photo, of that picture. Is it clear? So this is because the light wave is not having enough wavelength in order for you to make sure that it visualizes 2 distinct points of the cell structure. Now that is why we now use an electron, because an electron can also have a wave according to the de Broglie's principles in physics. So both, if it is a wave particle duality, which means that a particle can also behave as a wave and vice versa. So an electron can be a wave, and the wave of the electron has a smaller wavelength compared to the light wave, and that is why you can visualize now 2 distinct points in the cell structure with an electron microscope. So actually you have a good magnification here, but we have a poor resolution. So you see the nucleolus, you see the nuclear cell, the nuclear envelope, you see the euchromatin, but since it has been excessively magnified, you cannot have a good resolution. Okay, so this part here is going to be the nuclear envelope. Still, you have the euchromatin, you have the nucleolus, secretory vesicles, rough endoplasmic reticulum. Here we have cysteine. So this cell has much of rough endoplasmic reticulum, so it's much more of a secretory cell, and this secretory cell is much more going to be used in the production of proteins. So this is now a light microscopic view of a plasma cell. So the plasma cell have a typical characteristic that they have a clock appearance of their nucleus. As you can see, the nucleus, you have multiple heterochromatin that has like a clock appearance, and then they also have a Golgi complex at their terminal. So that one is a typical appearance of a plasma cell. So these are other cells. These structures here are the secretory granules. So this cell here, it is much more of a secretory cell, and these secretory cells, you can say it's a secretory cell. Why? Because it's having secretory granules that can be stored. At the image above there, this cell also have much. You see here we have instead much of smooth endoplasmic reticulum, tubular cells, but there is no. You can also have some of endoplasmic reticulum because the cell, secretory cell, this cell can also produce proteins. Is it clear? So you have smooth endoplasmic, much of smooth endoplasmic reticulum, but you also have some rough endoplasmic reticulum, and you must have a Golgi apparatus. Like here, a Golgi apparatus here. Like here, we have the Golgi apparatus here. So this at the terminal part here, the lowermost part, you have the nucleus with the nucleolus, the nuclear envelope, and the urochromatin, heterochromatin. Then it's going to secrete all the elements inside the lumen. When all that is triggered, it's going to produce secretory vector that's secreted into the lumen. Now in the image that we saw just initially, it is much more of it's a secretory cell, yes, but that secretory cell is not going to secrete into a lumen, but secrete into a blood vessel. But that's much more of an endocrine secretory cell. So this is the diagram, the light microscope view of that egg. This now is an exocrine secretory cell. So this diagram shows the vesicles, as you can see here. These are the secretory vesicles here. So the nucleolus at the terminal portion that's going to secrete. So this now is a secretory cell, an endocrine cell that secretes. As you can see here, we have the Golgi complex. You can see the Golgi complex. You see the nucleolus. You see the euchromatin. You see the lysosomes. The lysosome, but here you can see the lysosome here are much. So you have 1, 2, even 3 lysosome. You can think that this cell, since it has 3 ribosomes, because it can be one that is having that is using phagocytosis. When you see much lysosome, like you can think of a cell that has much of phagocytosis. But in the converse view, if you in controversy, you also have to think that this cell has also much of smooth endoplasmic reticulum. Though this cell has many lysosomes, it also has smooth endoplasmic reticulum. So that's why the cell is produced from the adrenal cortex and is a secretory cell from the endocrine gland. So this is just a light microscopic view of those steroid hormone secretory cells. Then the next thing, this view now, you see in here, you see many mitochondria. So when you see cells like that with many mitochondria, you can just see, you can think of a cell that is much more going to have cells with many mitochondria like that. It's mean that cell that is going to use much energy. Example of cell that can use much energy, you have muscle cells, and you have also cells that is mostly used in active transport, like in the renal tubules. You have many mitochondria here. So this is why this cell is a cell from iron pumping cell from the renal tubules. So that is the light microscopic view of those iron pumping cell of the renal tubules. You are actually having a tubule here, and then you have cells which are located here. So this is the proximal tubules of the cell, and this is the collecting duct. So from here we are finished now with the first part with cytology, the cell structure. We are now going to go to histology. Thanks for your kind attention.